The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, and the best digital health product.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Greetings and welcome to this special Galien Foundation webinar, where we're going to be celebrating the best of the very best in life sciences innovation over the last 50 years. On behalf of the Prix Galien Awards Committee, it's my pleasure to chair this, which is the second of 5 webinars, in which a total of 30 nominated products will be presented in preparation for what's going to be a truly landmark event, namely the Prix Galien Golden Jubilee Awards ceremony, which will take place in New York on October 27th. There, we're going to announce the winners of what we consider the single best innovative product brought forward by our industry over the last 50 years in 5 distinct categories. So there'll be one winner in each category. And the categories are pharmaceuticals, biotechnology, orphan and rare diseases, vaccines, and medical devices and technologies. Today's session is going to focus on the biotechnology sector, where more and more FDA-approved drugs are emerging from this sector. Drugs in categories like cell or gene-based therapies. These are high risk, but high reward in terms of benefit to patient. So it's a great opportunity for me and my fellow panelists to kind of review and reflect on the gains that the industry has made in improving the human condition through innovation. And we'll shed light on some of the challenges that are faced in this kind of journey, going from bench to bedside, and discuss each of the nominated products in their own unique and ultimately successful way. And just before I introduce each of the panelists, I want to say that they are all winners. They all should be congratulated to get to this stage. So many products, 50 years. It's just a pleasure and honor for me and for the Galien group to be here amongst these real winners. The people who will be speaking, and I'll just mention their names and titles now, as well as the product that they're representing. Dr. Maura Dickler, who's Vice President and Global Head of Product Development for Breast and Gynecological Cancer at Genentech Roche, and she will be representing 2 nominees, Avastin and Herceptin. Dr. Eric Rubin, who's the Senior Vice President of Oncology Early Development at Merck & Company for Keytruda. Dr. Mark Rutstein, who's Vice President, Oncology Development at Bristol Myers Squibb for Yervoy. Dr. Jochen Saalfeld, who's the Vice President, Immunology and Virology Discovery Research for AbbVie, and he'll represent Humira, and Dr. George Vratsanos, who's Vice President, Translational Science and Medicine at Janssen Immunology Therapeutic Area, Johnson & Johnson, speaking about Remicade. I also want the audience to know that we've allocated some time for questions at the end of our panel discussion, and you can submit your questions anytime while the webcast is going on using the Q&A button, which is at the bottom of your screen. And there's also going to be an audience poll conducted during the webinar, and that will cover during the webinar, in which you'll have a chance to actually vote on who you think, which product you think should be the winner. What we're going to cover today are two central themes. What's biotechnology innovation accomplished over the last 50 years using these exemplars in making medicines better, more useful, and importantly, accessible to patients with an increasing range of specialized needs. And then the second area is we're going to look forward to the next few decades, maybe the next 50 years, in terms of what new platforms are going to succeed the current wave of monoclonal, CAR-T, checkpoint inhibitors, et cetera, which just a few years ago were therapeutics that were never on the planet before. And we know that there are many more on the wings. So the way it's going to work now, we're going to move right into our discussion. And I've asked each of the company representatives to briefly address in no more than 5 minutes, just one of the following three questions, or if they have time to do more than one in that allocation, that would be fine. But they're to select one of the three. And the three questions are, why did your company choose to focus on the disease state and indication associated with the nominated product? And how significant was the level of unmet medical need when you made that selection? Two, is there a measurable impact that you can cite on clinical outcomes for patients with this product? What's been the clinical response to the product to date? Are there additional incremental advances still to be had with this product? And then thirdly, what challenges did your company confront in transitioning from early proof of concept to human subject testing and ultimately to market authorization? Are there lessons from your journey that might add to better public understanding of the medicine innovation process? Let's move on now to the five candidates, and I'm going to take them in order of alphabetical order of our panelists. And so I'd like to begin with Dr. Maura Dickler, who's going to speak first about Avastin and then Herceptin. Maura, please.
Yeah, thank you, Dr. Rosenblatt. And Roche and Genentech are honored to be nominated for both Herceptin and Avastin for this prestigious award. Really, Genentech was part of the birth of the biotech industry that occurred in the 70s and 80s in the San Francisco Bay Area. And really, as part of that transformation, Genentech scientists revolutionized the way cancer was treated with the development of Avastin and Herceptin, which were really some of the first therapeutic monoclonal antibodies to treat patients with cancer. So not only alphabetically am I first, but my drugs actually really are the oldest and really were paradigm changing at that time. There was a tremendous unmet need with really chemotherapy as being the mainstay or really the only way to treat cancer at that time. And, we were really desperate for innovation. For Herceptin, really, Herceptin is a HER2 targeting monoclonal antibody, and HER2 positive breast cancer, it's a subset of breast cancer, was recognized at that time as a very aggressive form of breast cancer, very poor prognosis, high likelihood of quick progression and death from that cancer. And really, Herceptin was the first HER2 targeting monoclonal antibody that really revolutionized the treatment landscape of this previously deadly form of breast cancer. Herceptin was initially approved in 1998, first in combination with chemotherapy, and initially first tested and approved in women with HER2 positive metastatic disease. And over many years, we tested Herceptin in women who had early stage breast cancer. And now Herceptin in combination with other HER2 targeting agents is really the mainstay for treating both metastatic and early stage HER2 positive breast cancer, and has significantly increased the number of women who we can cure with this previously deadly form of breast cancer. Avastin, another monoclonal antibody, really its discovery was based on the understanding of a protein that drove angiogenesis, and that all tumors need blood vessels to grow. And there was a protein that was really pivotal for that, and it's called VEGF, or human vascular endothelial growth factor. And Avastin is a monoclonal antibody that targets VEGF. And it was really discovered and developed within the walls of Genentech by Napoleon Ferrara in the 80s. Avastin really has been tested in a wide range of tumor types. It was first approved in 2004 by the US FDA to treat metastatic colorectal cancer, becoming the first anti-angiogenic therapy that was made widely available. And it's now really FDA approved across a number of tumor types. So I think I'll turn it back to you.
Oh, that's terrific. Thank you very much, Dr. Dickler. I think we're going to hold questions for the separate sections. So I'm going to move on now and ask Dr. Eric Rubin to speak about Keytruda. Eric, please.
Thanks, Dr. Rosenblatt. It's impossible to overstate the impact immuno-oncology therapies like Keytruda have had on the treatment of cancer and the lives of patients and their families. Cancer is one of the greatest challenges of our time. It's a complex and formidable enemy. At Merck, we are driven by a shared vision to invent a world where cancer isn't just treated, but cured. For us, that starts with Keytruda. Keytruda has fundamentally changed the way we think about cancer. The practice-changing impact of Keytruda across some of the most deadliest and prevalent forms of cancer is unmatched. Keytruda's story is as much a story about perseverance and teamwork as it is about challenging the idea of what is possible. Early in its clinical development, a group of us met to discuss how to prioritize potential Keytruda clinical studies. Dr. Roger Perlmutter, president of Merck Research Laboratories at the time, was asked where to draw the line based on resources. Dr. Perlmutter replied, "There is no line, do them all." In oncology, there is no line. Nearly 20 million people will be diagnosed with cancer this year alone. So as long as there are patients fighting in hospitals, families worrying in waiting rooms, and oncologists anxious to give good news, there is no line. We have adopted that ethos as a means of challenging the status quo and pushing the boundaries of what we think is possible. Our goal is to extend the benefits of Keytruda to as many patients as possible, including moving into earlier stages of disease where the potential for cure is greatest. I want to start by talking about Keynote-001, an innovative and seminal study and the bedrock of our clinical development program. It began in 2011 as a small dose-finding first-in-human study planned to enroll 35 patients. We started receiving calls from excited investigators reporting dramatic responses in their patients with metastatic melanoma. These were patients who had failed everything and now saw their tumors melting away. As a result of these early data, the FDA granted Keytruda breakthrough therapy designation for advanced melanoma in 2013. Keytruda was the first cancer drug to receive this designation, the first of many firsts for Keytruda. Based on impressive incoming data, Keynote-001 was adapted and expanded and ultimately enrolled over 1,200 patients, becoming the largest phase 1 study ever conducted. Keynote-001 led to multiple regulatory achievements, including the initial approval of Keytruda for advanced melanoma in September of 2014 which was the first approval for an anti-PD-1 therapy in the United States. Subsequently, the initial approval of Keytruda in metastatic small cell lung cancer in PD-L1 positive patients was achieved on the basis of this study in 2015, along with the first approval of a companion diagnostic for PD-L1 tumor expression in lung cancer. From the start, a key pillar of our strategy was to maximize our understanding of Keytruda monotherapy across cancers using innovative clinical trial designs and precision medicine tools. We used the PD-L1 biomarker to identify those patients most likely to benefit, while also recognizing that ultimately for some patients, a combination approach was needed. Our pursuit of PD-L1 as a diagnostic in lung cancer reflects our strong patient focus. Our biomarker strategy led us to evaluate novel combinations incorporating chemotherapy, culminating in the practice-changing results from KEYNOTE-24 and the KEYNOTE-189 studies. Before 2014, the 5-year survival rate for patients with metastatic lung cancer was 5%. This was essentially, in effect, a death sentence having this cancer diagnosis. In the first-line setting, Keytruda monotherapy increased 5-year survival rate 6 times over historic pre-immunotherapy norms for lung cancer patients with higher PD-L1 expression. 32% of patients still alive at 5 years. That's unprecedented. Today, 8 of lung cancer patients are treated with Keytruda based on the strength of our clinical data and dramatic survival benefit. As pioneers in the field of biomarkers, we are proud that Keytruda has 14 biomarker-driven indications and 2 tissue-agnostic approvals for MSI-high and TMB-high cancers, both the first of their kind in oncology. I'm struck by the fact that we are still so early in the Keytruda story. There is still so much more progress and impact to be made. Our clinical development program grew from 1 study in 2011 to now more than 1,600 ongoing clinical trials. In 8 years, we have secured 46 regulatory approvals in the United States and anticipate more than 80 potential additional new approvals over the next 5 years. Keytruda is now approved for 16 different types of cancers, including MSI-high and TMB-high cancers, the most approved uses for any targeted oncology therapy. I'm proud to say that more than 1 million patients have been treated with Keytruda worldwide in the commercial setting, and by the end of 2024, we plan to reach 2 million. Merck is rapidly advancing Keytruda into earlier stages of disease when many cancers are considered most treatable and potentially curable. To date, we've announced 6 positive pivotal studies for Keytruda-based regimens in earlier stages of cancer, the most of any cancer immunotherapy. We are also exploring innovative dosing and administration methods. Patient by patient, study after study, Keytruda has continued to transform the treatment of cancer and redefine expectations about what is possible. Despite the progress we've made, we recognize that there is still more work to be done. What we have achieved so far motivates us to work harder and faster because patients are waiting. We will follow the science to harness the potential of Keytruda because there is no line. Thank you, Dr. Rosenblatt.
Thank you, Dr. Rubin. Let's hear from Dr. Mark Rutstein, who's the Vice President of Oncology Development at Bristol Myers Squibb, and will be speaking on Yervoy.
Thank you so much. And on behalf of my colleagues at Bristol Myers Squibb, I'm extremely happy to be here and humbled to actually be in a discussion of such drugs of innovative achievement. And so I'm here to talk about Yervoy, CTLA-4 antibody. And indeed, the story of Yervoy is really one of pioneering. Immunotherapy has certainly transformed the way we think about treating cancer, deeper understanding of disease biology, and has brought therapeutic benefit to diseases with unmet need in a manner that we've never really seen before in the case of various disease states. So Yervoy was brought forth and first approved in melanoma as a monotherapy immunotherapy agent back in 2011. And at that time, actually, it had been 13 years since we'd seen an effective drug in melanoma. Indeed, at the time, metastatic melanoma had a median survival of around 6 months, and really an extraordinarily difficult disease to treat. Now, immunotherapy had not yet come on the scene at the time that Yervoy was being developed, and so indeed, BMS was a major pioneer in this setting. And the first studies with Yervoy and melanoma demonstrated significant advancement of the field, with median overall survival in the metastatic melanoma setting up to 10 months from the approximately 6 with the standard of care dacarbazine, and had shown a benefit as well in the resectable melanoma setting and the adjuvant setting, including an overall survival improvement. BMS had certainly gained a lot of experience in the immunotherapy space, leveraged that experience in the understanding about the interaction between the immune system and the tumor to hypothesize that combining the PD-1 inhibitor Opdivo with the CTLA-4 inhibitor Yervoy may actually promote greater benefit than either drug alone. And indeed, we believe at the time that there would be a very complementary mechanism of action. So in fact, in melanoma, there was the approval of the combination of Opdivo and Yervoy in 2015. At ASCO in 2022, we presented the study Checkmate 067, which is the combination of Opdivo and Yervoy in frontline melanoma. And I'm proud to say that at 7.5 years of minimum follow-up, the median survival is 71.2 months. Now, moments ago, I told you back in 2011, the median survival was 6 months. So a substantial improvement in the clinical benefit of these patients, truly transformational, unremarkable, unprecedented. And in fact, at the 7.5-year mark, approximately 48% of patients are still alive. So this combination of PD-1 and CTLA-4 has gone on to result in approvals and demonstrate durable survival benefits in renal cancer, in esophageal cancer, in mesothelioma, clinical benefit also in hepatocellular cancer and colorectal cancer. So durable benefits in diseases that had previously been very difficult to treat. And in terms of continued understanding of the CTLA-4 mechanism of action, there are a number of studies still going on, being run by BMS and others, including with our LAG-3 inhibitor, relatlimab, in various solid tumor disease states. And in addition, we've seen the development of and the proliferation of next generation CTLA-4 blocking agents, including the efforts to optimize the benefit risk profile of these assets of what we've seen with ipilimumab. So I would close by saying that the development of Yervoy was a pioneering step, a giant first step to usher in an era of immunotherapy. And we've seen with that giant first step transformational benefits in a number of solid tumor disease states. And this has been a journey that's been really started for immunotherapy in the 1890s. So one giant step forward and more to come. And thank you for your time.
Thank you, Dr. Rutstein. And in case anyone doubted that everything in medicine is immunology, this panel proves that we're going to move now from immune oncology to immunology and inflammation. And I'm going to turn to Dr. Jochen Saalfeld from AbbVie to speak about Humira.
Thank you, Dr. Rosenblatt. It's a really true pleasure to be here. Let me start with expressing my great appreciation for the innovation behind all the molecules we're discussing today. All of us share the same desire, offering treatment options for patients. Coming to Humira, let me state up front that everybody listening to this webinar should refer to the complete prescribing information for Humira at humira.com. All the historical and other aspects I will discuss are never intended to relay information inconsistent with that label. Importantly, follow the procedures of your country. At AbbVie, we've been raising the bar in the treatment of immune-mediated inflammatory diseases, and I will use going forward the much shorter term IMID, transforming patients' lives and inspiring hope and confidence for a better today and tomorrow for nearly a quarter century. And remember, the diseases I will be talking about are chronic, with severe consequences for the life of the patient, where they impact primarily the joint, the intestine, or the skin, with other important systemic consequences. You see, there was a time when substantial number of patients suffered from severe joint damage due to the ongoing disease activity in rheumatoid syndrome, frequently ending in severe disability or even being bound to a wheelchair. There was a time when a patient with two disease pathologies at the same time as an example of the severe and very debilitating spinal inflammation called ankylosing spondylitis, and at the same time, a form of an eye disease called uveitis, may have tried another TNF inhibitor. That molecule may have worked for the ankylosing spondylitis, but not for uveitis. There was a time when patients with very severe and very debilitating, truly very debilitating skin disease called hidradenitis suppurativa were treated with medicines like antibiotics because there was nothing else that physicians had. And actually, it wasn't fully appreciated. This was actually a chronic inflammatory disease. There was a time when patients with severe intestinal inflammation, like Crohn's disease or ulcerative colitis, had to receive regular infusions. Every infusion reminded the patient that they were sick and also impact the busy lives of often younger patient populations. All that changed when Humira, a therapeutic human TNF antibody, came on the scene. Tumor necrosis factor, or TNF, plays a major role in many of these IMiDs. And various attempts to inhibit TNF were made in the field, including anti-TNF antibodies or agents that had originally been tested for sepsis. However, none were customized solutions like Humira. From the beginning, Humira was developed with the patient in mind, putting the patient and the physician in control. Not only was it unequal breadth in clinical impact across many disease states in rheumatology, gastroenterology, and dermatology, it had also a very convenient subcutaneous self-administration option, no longer requiring infusions. Humira is approved today globally in 16 indications across rheumatology, dermatology, and gastroenterology, and has been used to treat more than 1 million patients worldwide. It is the only anti-TNF approved for hidradenitis suppurativa. Humira is also approved for treating Crohn's disease and ulcerative colitis, two diseases for which another anti-TNF, which is approved for rheumatoid arthritis, does not seem to work, possibly due to mechanistic differences. All this points us to this unique characteristic of this molecule, Humira. Let me add this to our discussion this afternoon. I understand why medicines can be grouped into classes like TNF antagonists, but I'd like to remind each of us that this kind of grouping is very simplistic and scientifically unsatisfactory. It ignores the fact that each therapeutic is unique as a result of its design and unique mechanism of action. Each is being developed and reviewed by agencies on its own terms. With patient care, our North Star, we continue to innovate by enhancing comprehensive care for patients facing these hardest to treat IMiDs by new ways of making the administration more convenient and other educational tools. Humira is a very important medicine across the world for the management of IMiDs, but we are also driving innovation across our immunology portfolio. Let me close. We know from discussion with patients that the initial diagnosis of one of these lifelong chronic diseases is a shock to their life plan and is felt as losing complete control of their lives. Comprehensive and convenient care allows patients and their physicians to regain that lost control based on great science in biotechnology, pharmaceutics, and medicine. Thank you.
Thank you, Dr. Saalfeld. And now for the final presentation, I'm going to turn to Dr. George Vratsanos from Janssen & Johnson & Johnson to speak about Remicade, please.
Thank you, Dr. Rosenblatt, and thank you to the Prix Galien Awards Committee for the honor of the nomination. We truly are humbled. And I'd like to address the first question you asked, which is essentially why we chose to study the initial indication for Remicade, which was Crohn's disease, and then touch briefly on what makes us so excited about our potential as a scientific organization going forward. Core to our values at Janssen is a deep commitment to addressing unmet patient need. And that stems from a deep dissatisfaction with the current status quo in terms of therapeutic options. Crohn's disease is a very good example of that, and it was the main reason why we chose that indication. Crohn's disease is extremely disruptive to a patient's quality of life. It's a challenge for patients. The disease often starts in the teen years, often by the 20s as well, and it often results in what we call a march to misery. Patients often end up with chronic pain, disability, the need for surgery, sometimes multiple surgeries, and before Remicade and the subsequent biologics, that was the course of disease. And I'm proud to say that Remicade and future treatments have dramatically changed that course for patients. A second example would be in rheumatoid arthritis. And this is an example where I personally witnessed as a clinician the impact of Remicade. I trained in the late 90s as a rheumatologist. I took care of some of the first patients at my institution with Remicade. And of course, we typically used patients who had some of the most difficult to treat disease with currently available therapies. And some of those patients responded quite dramatically well to Remicade. And subsequently, it's been shown unequivocally that Remicade does have the potential, the benefit of changing the course of disease in a disease such as rheumatoid arthritis, where it's been shown to inhibit the development of erosions that end up disabling the joint and cause major disability. Over the last 20 plus years, this product has been approved in 16 indications. And when you total that, it accumulates to more than 2.5 million patients treated globally across a spectrum of immune diseases, touching on dermatology, gastroenterology, and rheumatology. And despite the fact that biosimilars are now available, more than 20 years after its introduction, it remains a very important therapeutic option for many patients worldwide. And the safety record is very supportive of its ongoing therapeutic use, and we stand behind that strongly. I'd like to point out, however, that despite the success of Remicade and subsequent biologics, the unmet need persists. Our goal is remission. It's quite simple. And in the group of immune-mediated diseases that we study within our therapeutic area, it's a fact that more than 90% of patients do not achieve what we would call durable remission. And that's what drives us today as a company. And I'm very proud to say and excited about the fact that our organization, including our Translational Science and Medicine organization, is better positioned than ever before to translate the latest scientific advances and current technologies into truly novel and compelling new therapeutic options for patients and approaches. So thank you for your attention, and I'll turn it back to you, Dr. Rosenblatt.
Thank you, Dr. Vratsanos. So what I'd like to do now is just go again through in the same order, asking each of the panelists one question, the same question about the future. At the same time, either now or very soon, now that the audience has heard all of these presentations, I'd like to open up the voting mechanism for them. And at the end, we'll hear the results of the voting. So let me go now to the to the next round. One question that I'm going to have for all of you will go in the same order, and that is, where do you see the next wave of innovation in your field, the field that you were representing? What are the new areas of science that excite you the most? And if you fast forward and imagine you're sitting on a Galien review panel for awards 5 years from now, where do you think that biotech therapies are going to be? This is a chance to just talk about predictions for the future. And Dr. Dickler, let me turn to you first.
Yes, sure. And you were coming in and out. I don't know if that's on my end or some technical issue.
Oh,
also.
Is that true of everyone?
Yeah.
Yep.
But I think I understand the question. I just wanted to, in terms of our technical difficulties, Well, I think building upon the drugs that are within this session, which are really, generally monoclonal antibodies that really have been developed to treat disease, and mine being, therapeutic monoclonal antibodies in cancer. I think some innovation that has already occurred is that we were able to conjugate cytotoxic payloads to some of these antibodies to create antibody drug conjugates, and some of these already exist. Herceptin or has had a payload conjugated to it, and that's Kadcyla, and that's effective in HER2 positive disease. And there really is a new wave of innovation in cancer to leverage these monoclonal antibodies as ways to deliver payloads. And I think one interesting area that we're going to start to see a lot of innovation is the types of payloads that we deliver. For a while, it took us time to get those antibody drug conjugates right, in that sometimes there were problems with the linker, and so the cytotoxic payload was released early and caused toxicity. And I think we're seeing major advances such that our delivery of the payload, and generally now chemotherapy to cancer cells, is much better, so that the therapeutic window is narrowing, and we're really able to get it right. And there's many drugs coming down the pike where payloads will change. Some of them may be immuno-oncology agents, where we rev the immune system, and others might be protein degraders. And I think that there's a lot of really interesting work in that area. Also building a little bit on just immuno-oncology in general, we've heard a lot about a number of monoclonal antibodies that have helped us to harness the immune system, and I think we'll continue to see a lot of innovation on that front. I would love one day to see vaccines able to stimulate the immune system, and I think one day we will be able to do this so that we could treat patients who are potentially cured from cancer. Let's say they've had surgery and we want to treat microscopic residual disease, and I think that vaccines are on the horizon, and something aspirational, but I feel within our reach. So those are some of my thoughts. Thank you.
Great. Thanks for that look into the future. And Dr. Rubin, tell us what you see.
Sure. Thanks, Dr. Rosenblatt. So I want to second what Dr. Dickler mentioned about sort of what I might say as add-ons to monoclonal antibodies. Again, it's perhaps not surprising that all the candidates today are monoclonal antibodies. And I think the field is now moving to sort of next generations that include conjugations, as Dr. Dickler mentioned. I think many of us were at ASCO a couple months ago, where there was a standing ovation at the plenary session. That doesn't happen too often, when the results of a HER2 antibody drug conjugate, trastuzumab deruxtecan, was presented. And in part, this was because it really was redefining what's considered a HER2 positive breast cancer patient, enabling an effective treatment to reach many patients who would historically have been characterized as HER2 low and not been able to get Herceptin, Dr. Dickler's drug. And there are many of these. I think it's a large area of investigation right now. It's quite exciting. Lots of new targets, lots of new payloads, and so I do think seems to be the next wave, is antibody drug conjugates. And there are related approaches such as bispecifics or trispecifics that are, again, bringing things together with a targeted monoclonal antibody. The second area I'll touch upon is diagnostics. Again, I think this is increasingly important in enabling effective treatment for cancer patients. I think I'll comment. I think one that's emerging is as a result of advances in DNA and RNA sequencing, we now have very sophisticated and sensitive methods to assess circulating tumor DNA in blood. And I think this is likely to have a large impact not only on trial design and ways to identify patients at risk and enable more rapid development of therapeutics, but also to understand mechanisms of resistance, since it becomes much easier to obtain sequential liquid biopsies, if you will, for patients on treatment.
That's great. Thank you. Dr. Rutstein, your thoughts?
Thank you. And I certainly wholeheartedly agree with the comments from Drs. Dickler and Rubin, and would add maybe a few additional areas. And one of the areas that's of strong interest to Bristol Myers and others is in the field of protein homeostasis. So looking at protein degraders. And indeed, if we think about traditional oncology therapeutic agents, many of them are involved with pathway inhibition. And of course, mutation can occur, resistance can occur, and therefore that even though pathway inhibition, as evidenced by conversations of drugs today, is very effective, it can be limited in the fact that only approximately 10% of proteins could be addressable. Proteins involved, let's say, in disease, in oncologic disease and other diseases, could be addressable by conventional means. And so one of the things that our organization and others is focused on is what we call harvesting or leveraging the proteasome ubiquitin pathway and looking at protein degradation to potentially expand the repertoire to thousands of more proteins previously undruggable, and to uncover druggability. And indeed, there are a few types of assets in that arena we call cell mods or ligand-directed degraders that specifically degrade proteins that are involved in disease pathogenesis. And there are a number of these assets in the clinic. And again, it's making the undruggable druggable that is very attractive for this type of agent. I would also add, in addition, we've seen remarkable advances with CAR T and cell therapy, and certainly adaptations and next wave of CAR T are envisioned, including looking at CAR Ts or cell therapy, CAR T mechanism where more than one antigen can be addressed, therefore addressing the potential of antigen loss for the initial CAR T, or attaching a payload to the CAR T, as an example, to overcome resistance. And of course, people often talk about the entree of CAR T into the solid tumor arena beyond the hematology space, where pervasive benefit has been shown. And that would potentially require an allogeneic off-the-shelf approach. So certainly a lot to think about in the areas of protein homeostasis and evolution of the next wave of CAR T cell therapies, along with, again, what was already stated in terms of ADCs, bispecific antibodies, NK cell engagers, T cell engagers. There's a lot to look forward to. Thank you.
Thanks for those additional views. Dr. Saalfeld.
Thank you for the question. I agree with the other speakers. We at AbbVie are exploring ADCs in the immune space. That's something that we are interested in, just to explore how well that works. I do agree with others, Dr. Rutstein, about looking at new platforms. But I want to come back to what Dr. Rubin said about diagnostics. I think in the immune space, one thing that we need to learn over time is how to diagnose patients much earlier and then determine which patient is the right patient for the right medicine. I think oncology is a step ahead in this, and we need to learn from how oncology is doing this. The immune system is very complicated, and to have actually precise assays that can actually select patients for the right treatment will be a challenge. But I believe we have to go in that direction to really, at the end of the day, give the patient and the physician more decision support tools to choose the right medicine that's right for them. I think the other area, and Dr. Sanos started talking about that, is the whole question of cures. Again, if you can identify patients much earlier, understand the disease mechanisms, and use some of the sort of emerging insights around reestablishing homeostasis, is actually a way to achieve cures in autoimmune diseases. So those are the areas that I would be looking for as innovation in the space I discussed in my remarks today.
Terrific. Thank you. Dr. Vratsanos, please.
I echo my panelists in their excitement about the future of science in oncology and in immunology. We too are very excited by the potential for multiple platforms. I want to highlight one new avenue for us, which is the opportunity using data science to generate insights that have not previously been available through conventional techniques. And at Janssen Immunology, we're very excited by the possibility of using those computational methods to do what Dr. Solfa just mentioned, which is to identify the subset of patients within a common disease who will benefit best from a therapeutic. Second, to identify novel markers of disease that would enable more innovative clinical trials to be conducted. And third, I'll highlight the possibility, and now we hope soon to be reality, of doing combination approaches to reach a much deeper level of response than what we've historically achieved. I'll highlight a recent trial we conducted, which was a phase 2 study in ulcerative colitis, now published, which used a combination of a TNF inhibitor, golimumab, plus our IL-23 inhibitor, Tremfya, to achieve roughly double the remission rate for patients with UC than either treatment alone. And I think our future will be to do more and more of these types of studies, where, of course, the biology is complex and we'll need at least 2, potentially more approaches to really get to the deeper level of response we all aspire to. So with all these factors underway, we're very excited by our potential going forward as a research and development organization to continue to innovate in the ways that our company and the other companies highlighted today have already done.
These are terrific views of the future and perspectives. I wanted to ask an open question to the group. Does the way that you think about development or regulation, has that changed as a result of these pioneering drugs changing the development and regulatory paradigm, or the pandemic increasing the speed of regulatory agencies? On the one hand, I could imagine that might accelerate things. On the other hand, we have modalities that have never existed before that, the regulatory agencies don't necessarily have a lot of comfort with. Do people have thoughts on how that influences what you're doing now for drugs that are going to be therapies that are going to be emerging 5-10 years from now?
So Dr. Rosenblatt, maybe I'll just chime in because I briefly mentioned this. I think one of the areas that's been quite interesting, and there was just another one that happened recently, is this idea, you might call it sort of redefining how one classifies cancer. In the early days, we classified cancer because the treatments were basically surgical and radiation therapy. So it was always the tissue of origin, even though the cancer was metastatic. And as our tools and diagnostics have improved and we understand pathways that are involved, we realize that there are many of these pathways go across cancers, and this enables thinking of a pan-tumor approach, especially for drugs that are targeted around those particular alterations. And there were some efforts to do this for a while, but the first one, again, honored to have the first one, which was an MSI high pan-tumor for Keytruda, based upon really great science, from Hopkins investigators, where they recognize these patients should be generating lots of new antigens that could be recognized by the immune system and should be highly responsive to anti-PD-1 drugs, and they were right. And I think, again, I have to point to the FDA, I think, has been quite innovative in supporting such a view, because this required quite a bit of innovation. You can't do randomized trials in a cancer type where less than 1% of the population of the cancer has that genetic alteration. It's just not possible. But they've enabled pan-tumor approvals, and there are now, I think, 4 of them, with the last one being BRAF/MEK, I think last week. So I think that's, to me, a quite notable accomplishment for the field from a regulatory perspective.
Perfect. Other thoughts?
I would maybe add that with some of the oncology therapeutics that have come through, and some of the discussions that we've had with partner regulatory agencies, the issue of endpoint surrogacy has become an interesting topic, right? So when we think about accelerated approval paradigms in order to bring forth drugs to address unmet needs as soon as possible, when are surrogate endpoints of efficacy of a drug sufficient, and effect sizes and surrogate endpoints sufficient to support accelerated approval. And then there have been further discussions with agencies around even novel surrogate endpoints. And more recently, we've seen also discussions on minimal residual disease in the solid tumor setting, which has typically been in the hematology setting, and the potential adaptation of minimal residual disease to the solid tumor setting, and the use of measurement of something like circulating tumor DNA, for instance, to select patients or to establish endpoint surrogacy in early stage oncology settings, for instance, in adjuvant trials. So I think endpoint surrogacy is becoming an increasingly important topic and a good topic for partnership with regulatory agency.
Other thoughts?
No, I agree. I think also another area that's really very interesting is DNA damage response. And I think that there have been some drugs, such as the PARP inhibitors, PARP1/2 inhibitors, that have been effective initially in a narrow group of patients that had inherited germline alterations, and more recently broader in all comers in ovary and prostate. But I think what we're seeing there is there's a lot of ongoing learning in the lab about new proteins that are important in how we repair damaged DNA. And as we learn more about these mechanisms, we're also able to create drugs that can potentially inhibit them. So I think that there's going to be an explosion in drugs in this DNA damage response space, where we'll see a whole lot of new agents. And I apologize that my cat is joining the panel. It's a problem with working from home. I think it's going to be a really fascinating area. And again, not just for patients with inherited forms of cancer, but even more broadly as we really better understand how to use these drugs.
Terrific. Thank you. Does anyone else wish to comment? And if not, I'll move to the wrap up now. Terrific. Well, what a terrific discussion. And I just want to say a few words in closing. First of all, we'd love to have the poll taken now, and then we'll be able to give you the results in a few moments. But I really, so take a look at it and please vote. And I just want to thank all of the company representatives who are here. This is a terrific, thoughtful panel. As I said earlier, everyone here is a winner. Those of you who want to watch this webcast, it's going to be viewable through October on the Galien Foundation's homepage, which is www.galienfoundation.org. And I want to thank everyone, wish everyone luck, and I look forward to seeing everyone in New York in person on October 27th for the ceremony and the dinner. So let's see this. I think, yep, I'm going to assume everyone has voted, and I am now going to close the poll. And you can see the results here. Is that visible to everyone? With Keytruda in the lead and Yervoy is second. This is a sort of an opinion poll. This will go to a formal awards review committee. So again, I want to just thank everyone for this terrific panel discussion. It's a pleasure to interact with all of you and an honor to interact with all of you. And my congratulations to all of you. Thank you, everyone. Bye-bye.